All right, let's continue on part two when we were talking about the immune system. And it shouldn't really take very long to kind of wrap this chapter up. We're going to look at categories of immune response, steps in inflammation, talk about different types of antimicrobial host-derived type things. We're going to talk about phagocytosis, how a fever actually helps throw the game towards our favor, and mode of action of the complement system. Okay, so the second line of defense, we have non-specific and generalized types of responses from the immune system itself, and these include phagocytosis, inflammation, fever, and antimicrobial type of proteins that are secreted. Phagocytosis, which is Bagum and Tagum in Recycle, surveys the tissue compartments and discovers microbes, particulate matter, ingests, and in the process of ingesting eliminates those materials, and extracts intelligence. In other words, it's interrogating, it's doing a washboard, or however you want to look at it, extracts immunogenic information. We call them antigens, and that trains the immune system. The type of phagocytes, we have neutrophils, monocytes, and macrophages. Neutrophils are general purpose, react early in inflammation response to bacteria. Usually you'll see an elevated neutrophil. Of course, a high count would mean that there's a bacterial infection of some kind, and it's the primary component of pus. The pimple popper doctor would probably know a lot more about that. Monocytes are transformed into macrophages after they migrate out of the bloodstream into the tissues. Histiocytes live in certain types of tissue. We have alveolar lung macrophages, Kupffer cells that are in the liver, dendritic cells in the skin, macrophages in the spleen, lymph nodes, bone marrow, bone itself, and the brain. Macrophages, interesting beasts. They have passes. They can go pretty much anywhere. They have this wonderful characteristic of changing their shape and size to skinny in between cells, and they're found in lung cells, Langerhans dendritic cells. You can see various places, but these they have behind the scenes access, a pit pass, I guess you could say. A phagocyte means phago, eat. O is a combining form inside a cell, so it's an eating cell. It's a physical process of engulfment, dismantling, recycling, however you want to look at it. It can be isolated event or part of an overall orchestrated event. Chemotaxis, ingestion, phagosome formation, destruction, excretion. It's sort of the main features. So it's attracted to it, eats it, breaks it down, and then excretes useful parts. And this goes into more detail, so this is probably a good table to review for a class exam. Chemotaxis moves to the site adhesion, used to recognize the PRRs. If we recall, we talked about those in the PAMPs, those specific receptors. And then we have engulfment and phagosome formation, and killing. So they're actually killing, as you can kind of see, as the process. And then we release things that can be recycled or just debris in general to be cleaned out by the lymph system or whatever happens to be available. So the PAMPs we talked about is pathogen-associated molecular patterns. These are signal molecules found on surfaces. These are sugar protein type arrangements. These molecules are shared by many organisms and not present in mammals. Serve as red flags for phagocytosis in other cells. So it's just one of those things. It's a unique barcode, I guess you could say. The bacteria PAMPs are peptidoglycan and liposaccharide. So it's a peptido is a protein and sugar, and lipopolysaccharides is a long strain. Viral PAMPs, a double-stranded RNA, can serve as a pathogen associated. The receptors in the host, found in phagocytes, dendrites, and various others, recognize and bind the PAMPs. Cells possess PRRs whether they have encountered PAMPs before or not. So the receptors are there, of course. PRRs are found within cytoplasm of phagocytic cells. They recognize the PAMPs once they've been phagocytized, and then releases signals and helps to regulate the inflammation that goes on from there. Certain things we want to look for signs. You're going to be drilled these sorts of things when you get more nursing. Rubor is redness caused by increased circulation and vasodilation. Calor is the warmth cause. So basically, you go into any retirement home and go feel an elderly person's shin, and you'll see rubor, you'll see calor. A tumor is swelling caused by fluid escaping into other tissues, and you see like a foot swelling, or it's getting bad as elephantiasis. Dallor is pain caused, and the swelling and expansion of the tissue makes it really painful, and loss of function. Inflammatory response. Some chronic diseases, cardiovascular disease, are caused by chronic inflammation. It causes damage, and the body tries to deal with that. And it's like having open lesions in your circulatory system. Some researchers believe that aging is a consequence of increasing inflammation. I can tell you that. Trauma from infection, tissue injury, or necrosis due to physical or chemical agents. Specific immune reactions. It could be triggered by just something as simple as pollen or something. Chief functions: to mobilize and attract immune components to the site of an injury. Sets in motion the mechanisms to repair tissue damage and clear away harmful substances, and to destroy microbes. I would have put that one at the top of the list, but hey, I didn't write the book. Inflammatory response is a powerful defensive reaction. It's a means for the body to maintain stability, restore itself. It does have the potential to cause tissue injury, destruction, and disease. The idea is you want to win the battle as quickly as possible, and if there's collateral damage, then that's going to be some. Process leading to inflammation is dynamic, is a predictable series of events. Once an injury has occurred, these reactions will, a chain reaction will start going on and start migrating. So here is the inflammation. You can see bacteria in the wound. You get vascular reactions, so you got damaged vessels, and various things have to be repaired. You get edema and pus formation, and then a scar formation. Try to heal and seal the best we can. Diapedesis, as I've already referred to that as without calling it by name. Dia means through, ped means walk or move, and esis is a process. So it's the process of moving across something. So the migration of white blood cells out of the vessels into tissues. The idea is they can change their shape and skinny in between cells, and it gives them the backstage permit. They have some characteristics that allow them to go anywhere in extracellular spaces. They're attracted to various chemicals or stimuli, and they move out to do that. And there's diapedesis. You can see it skinning in between cells, literally changing its shape, and it's responding to maybe a chemotaxic factor of some kind, some chemical that's causing it to be an attractant for it to move towards it. And quite an interesting process. Benefits of edema in leaky vessels: influx of fluid dilutes toxins that can form. Fibrin clots can trap microbes to try to contain it. Neutrophils aggregate at the inflamed site, involved in phagocytosing and destroying bacteria. Pus is the accumulation of that whitish mass. And pyogenic bacteria such as strep and staph and gonococcus and meningococci that stimulate the formation of pus. Of course, it's the resulting from wars. Fever, abnormally elevated body temperature, 98.6 is our normal. If you have a low-grade fever, it's anywhere to up about 101. High-grade fevers go from 104 to 106, which are very dangerous. You can get brain damage if it's allowed to go on. So a lot of times they'll start cooling, putting in ice and that sort of thing. You get that high. Substances that reset the hypothalamic thermostat to a higher setting are pyrogens, things that actually cause, pyro means fire, like a pyromaniac, like somebody likes to play with fire. So these are things that are on the outside, products of infectious agents such as viruses, bacteria, protozoan, like this virus that we're dealing with now, triggers a fever response. Fungi, endotoxin, blood products, vaccines, or injectable solutions. Endogenous pyrogens are liberated by monocytes, neutrophils, macrophages, things that are involved in first orders of battle. The idea is you increase the temperature and some protein will fail or become inactive as a result that are associated with the pathogen, and that's why it's an effective way of trying to deal with the infection. Benefits of a fever: inhibits multiplication of temperature-sensitive microorganisms, poliovirus, cold viruses, herpes zoster, systemic and subcutaneous fungal pathogens. And just remember, the fungal pathogens are ones that we have to deal with all the time. Impedes nutrition of bacteria by reducing the availability of iron. If you remember, I've kind of been harping on iron. It's all about the iron, charging transfer RNAs and that sort of thing. It's all about the competition for that iron, the host iron, and the bacteria need it. Increases metabolism and stimulates immune reactions and naturally protective physiological processes. Speeds up the ability to make some red blood cells, hematopoiesis, phagocytosis, and specific immune reactions occur. Benefits of a fever: make it difficult to treat or not to treat. The side effects: tachycardia, technopia, which is an elevated respiratory rate, and lowering of seizure threshold. And you can go into seizures, and sometimes they'll put somebody in a temporary coma to deal with this if it's long term. Antimicrobial products: interferon, small proteins produced naturally by certain types of white blood cells. Thought originally to be against viruses, but it's against pretty much anything. It's produced by lymphocytes, fibroblasts, and macrophages. Interferon, what we call gamma interferon, is produced by the T cells. The activity of this interferon is it binds to the cell surfaces and induces changes in the genetic expression, and all three of the interferons can inhibit the expression of cancer genes, tumor suppressor effects. They have alpha and beta versions that stimulate phagocytosis. It's a way to ramp up our. It's sort of like mobilizing with more weapons our troops. We're having a special type of weapon that can see around corners and things like that. Gives it a little bit more of an advantage. Binding of viruses and other microbes to receptors on host cells signals the cell to produce the interferon, and the binding of the interferon induces the production of proteins that inhibit viral multiplication. Usually we're after the RNA type, prevent translation of viral proteins, not microbe specific. It is a valuable treatment for virus infections, however. You can see nearby cells and the interferon factors the signals, and they're very potent signals to turn on aspects of our natural way of dealing with things. Complement. Now, when you hear the word complement, I want you to think of the word cascade of events. So it's like dominoes. You hit the first domino, it hits the next domino, hits the next. So it's cascading across, and that's really what we're doing with complement. Sometimes it's thought of, once the complement is started, it's really hard to stop the process, and so that's there's a lot of checks and balances before our bodies commit on this. But cascade reactions is a sequential, if we think of dominoes, physiological response. The first substance in the chemical series activates the next substance, which activates the next. So instead of just dominoes falling, it's a chemical reaction that causes the next process to be consumed as part of that switching. So without going into too much of the detail, there's a series of these proteins. C3 protein, either free or bound to a pathogen, is hydrolyzed into C3b and A. This activation, just cleaving of this protein, then causes C5 to go to C5a. You don't have to go all through this. I just wanted you to kind of see that it is just a cascading of events, and you break down a substance that now it has new active forms, and then it breaks down yet another protein, and it has active forms, and it goes on and on and on, and ultimately leading to various things to get rid of things that might be after us. We need to destroy them. So classical complement is initiated by binding to the antibodies that are already bound to microbes. So the FC portion, the constant region of the antibody, is sticking out when its antigen binding region, we'll talk about it later, but it's sticking out, so that's used to initiate complement. There are alternative complement pathways that don't need antibodies, initiated by the presence of just foreign cell antigens, and sometimes that response is sort of the shortcut to get the process started. So here's what I like for the image. This is really well done. You can see that C3 hydrolyzes and catalyzes into two components. One now cleaves C5 into C5AB, and then polymerization of the 5B, it goes to C6, 7, and all these sorts of, the idea is it goes and attacks the membrane and literally blows the cell apart. Once the process starts, it's really hard. Other products, short proteins, about 12 to 50 amino acids, defensin and others, able to insert themselves into the bacterial membrane. So what we're doing is poking holes. They say creating a pore. I say it's like putting one of those spike strips across the road. The police will do that to try to stop somebody running from them. It pokes holes in the tires. The same sort of concept is the membrane gets enough holes, the cell starts to lyse, and they can't stop it. That it's like they don't have access to fix a flat while they're trying to do things. So antimicrobial peptides disrupt membranes. That's all this is showing, that it's disrupting the phospholipid bilayer, which it's got hydrophilic and hydrophobic regions. So hydrophilic we see positively charged, and it's trying to get in. What ultimately it's doing is damaging the membrane and poking holes in it, and the bacteria can't recover from that. So that's the end of this series for this chapter. What I would do is to get an outline. Now I've posted a bunch of resources in the course resource area. There's videos that you can watch on the immune system. There are a ton of them. Instead of picking out one or two, because I don't want you to think of it as an assignment, if you're confused about some of the concepts that we've talked about, you can find a YouTube video on it, and that way I don't post a whole bunch for you to review to waste your time. I just want you to go after things. If you can't find a video and you need one, you just let me know, and I have a big collection of these things, and I'll be glad to do that. So be safe, and I will see you next week when we start going on to the next chapter, to continuing with the immune response, which is really, really important.